The reason why I picked this is because they provide this great foundation for ecosystems that often goes unseen and unappreciated. Microbes are not as charismatic as cute animals, or even, I would say, cute plants. So we often don't think about them when we're looking out at the landscape of an ecosystem. So that's why I picked this topic, but let's jump to who. If I can move my slides. Okay, there we go. So when we think of fungi and mushrooms, we often just think of the part that we can see above the ground occasionally. Or it might be sitting on your pizza or in your pasta dish. This is just one part of a fungus, and not all fungi produce mushrooms. And this part is the part that grows when they're trying to disperse their spores. It comes out of the ground, but the vast majority of the fungi's body is actually under the ground, forming these thin filaments like plant roots. They're like little threads under the ground, and that's where most of the fungi is found. So this is often what we think about. But the most interesting things that fungi do are often microscopic, so we can't see them. And to give an example of this, I want to jump to fungal spores. And fungal spores kind of do the same things that seeds do for plants. They're basically how the fungi reproduce and travel to new areas, and they're often really tiny. So this is actually an image of Aspergillus spores. Aspergillus is a really common fungi that's found not just in the environment, but in your homes. So it's kind of everywhere. So this is an electron microscope image, a very, very tiny image, spores kind of on the end of the little filament that I was talking about. And there's a lot of diversity in these spores and their sizes, their shapes, what they look like. And the diversity of these sizes and shapes often contribute to how far they can travel in the air. So whether they can travel a lot further, how tolerant they are to stress. So if you imagine being a tiny spore traveling thousands of miles in the air, you're exposed to sun constantly, right? There's less UV protection higher up in the atmosphere. So you're roasting out there, there's no water in the atmosphere. So spores have a lot of different characteristics between species of what makes them best at dispersing. And that's part of how they rule the world, is they're able to get really, really far. Spores can travel a wide variety of distances depending on what types of mushrooms they come from. So some mushrooms just drop their spores within a few feet. Others, their spores travel maybe 10 kilometers, so they're kind of staying within the same ecosystem. And then other spores, like these small ones, can travel thousands of miles, so across oceans, across continents, and they're still viable when they land. They're still alive. They've made it through that really, really tough journey. So how fungi disperse, how they get there, if they can survive, these are all really, really important characteristics that allow them to rule the world. So I'm going to talk about how these fungi move around, and then I'm going to talk a little bit about those functions they perform in ecosystems that contribute to a lot of different things we see. So first, how do they move? Some spores have really active mechanisms of propelling their spores out. They build up pressure and eject those spores. Other fungi rely on rain falling on them to kind of push those spores up into the air. The ones that travel a lot further, and the ones that travel in drier areas like Southern California, a lot of spores in Southern California aren't relying on rain to get around, otherwise they wouldn't get very far. So another mechanism that is kind of more relevant to where we are now is fungi growing in the soil, like I mentioned earlier, can actually be lofted into the air with dust and soil, especially when it's dry and windy. So inside the soil, because you can't see it, I have this image under a microscope of these fungal spores that look kind of like little cans. They're little barrels, and you can see little thin filaments connecting them. So these spores are actually designed to break apart between each of those little mini cans. And that makes them smaller and lighter so they can travel a lot further in the air. To give you well, it's not the best way to say it, but to give you an idea of how small they are, it's 10 microns, so pretty tiny. You can't see them even with a lot of microscopes. And now I've been alluding to how they rule the world and all the functions they play in ecosystems. I'm going to go back to this diagram a lot. So if you're like me, I have a hard time paying attention to things for a long time, but pay attention to this part because I'm going to use the icons throughout the rest of the presentation. So fungi play three general, really important roles in ecosystems. The first one is pathogens. And we don't often think about pathogens as a role in ecosystems, but what they do is they actually improve biodiversity. And I'm sure most of you know that greater biodiversity is usually a good indicator of a healthy ecosystem. So part of the reason biodiversity is maintained, because you can imagine with two things competing against each other, the one that is better at doing that is always going to win. However, whatever organism that is still prone to being infected. So with really low levels of pathogens in an ecosystem, you can kind of give a step up to other organisms that might not do as well in other conditions. So low levels of pathogens help biodiversity. Something I'll touch on later though is if you have more pathogens in the ecosystem than what you're really supposed to have, it actually have reduced biodiversity. So the role of fungal pathogens, I'll touch on again after this, but most people don't think of that as a force structuring ecosystems, but they are important. The second group are decomposers. So these are the fungi that break down dead material in the environment. Bacteria, maybe I'm jumping too much into the microbial world. Bacteria get like a lot of credit for this, but I'm just gonna say that fungi actually do a lot of the heavy lifting with decomposing dead material. The reason for that is a lot of plants and trees contain this material made out of carbon that's really tough to break down, super tough. In fact, it's so tough that no bacteria can break down this material, but fungi can. So a lot of the carbon compounds that need to decompose and need to break down, they're kind of locked into this tough material. So fungi do the first step by breaking down that tough material and creating better access for other decomposers like bacteria to break down dead materials. So they're kind of like nature's garbage people. They take out all the dead material. If we didn't have fungi, you would just have dead vegetation kind of piling up and choking everything. So they are super important in the environment. Decomposition releases carbon dioxide. That's a natural cycle in the environment. Humans, we also release a lot of carbon dioxide with our activities. And what's interesting is it's often talked about how our carbon emissions contribute to climate change. They definitely do. The balance is so delicate. It's really interesting when you think about the carbon dioxide that's produced from decomposition, mostly by fungi. It's quite a few times greater than what humans produce, period, but the amount that we produce just adds on to that really delicate balance. And I'll talk more about that in a little bit. So we have pathogens, we have decomposers that break things down. They also produce carbon dioxide when they do that, and we'll touch more on that later. The last group are the symbionts, and this is just a word that means organisms that have a close-knit relationship with other organisms. So often they rely on each other to grow, to live. Symbiotic fungi, maybe some of you that work in pulling invasives or in the greenhouse or in the garden by the lighthouse know this, but mycorrhizal fungi are really important to plant growth. And mycorrhizal fungi are essentially an extension of plant roots. So they connect with the plant roots. Some of them actually grow in between the cells of the plant roots. Some of them just encase plant roots. And they act as this extension so that the plant can get more water, it can get more nutrients because it's able to cover a larger amount of soil. So if you pull up plants in the garden or the greenhouse, or you're pulling up invasive plants, sometimes, depending on where you are, you might see long, thin, white roots. It may be plant roots, or it might actually be mycorrhizal fungi. So this mycorrhizal fungi, it's found in the vast majority of plants you see. I would say between 70 and 80% of plants rely on mycorrhizal fungi to get enough water, to get enough nutrients. The fungi actually protect them from disease and sometimes heavy metals, depending on the species. So symbiotic fungi do a lot of good for plant growth. And I just want to point out that plant growth is a way of capturing carbon from the atmosphere. So kind of an opposing force to decomposers. Decomposers are releasing carbon dioxide when they're decomposing, but symbionts are helping plants grow and kind of capture that carbon dioxide from the air. So these three groups are going to be the main characters in the story I'll tell. So red pathogens, decomposers are blue with the little cubes kind of breaking down into smaller pieces, and then symbionts help plants generally. So I had alluded to climate change earlier, and I just want to kind of give context for this entire presentation. It's one of the biggest challenges a lot of us will face in our lifetime. It's happening now. This map shows the globe, and it shows these unusual temperature differences that are persistent. So you'll see there's a scale as you go into that darker red, that's a 5 degree Fahrenheit difference from average. And this is a lot of data that's been compiled into this. And you also have blue areas where it'll become colder. So where we are in Southern California will be quite a few degrees above average. So we're gonna see warmer and drier conditions in San Diego as we move forward in time. We'll have actually similar amounts of rain when you look at the total, but rain events are going to start to become more sporadic. And if they're becoming more sporadic, but we're still having the same amount of rain, these rain events are going to become much, much more intense. So some of you will see when the park reopens, there's damage on our trails. And that comes from parts of the trail being washed off after rains. So under climate change, we might see less rain, so the ground is a lot drier. And then when it does rain, that rain's just gonna start carrying away large parts of the trail or just washing them away because it's much more rain on a drier trail. So just giving context for this presentation and for the park, this is one of the biggest challenges. And it's important to understand how this might change the types of fungi we see in ecosystems, right? Because if these fungi are pathogens that have impacts on biodiversity, if they're the decomposers that are breaking down our dead material but releasing carbon dioxide, and they're also the fungi that are helping with plant growth, we're going to want to know how these fungi are going to react to climate change and what that means for the ecosystems in turn. So wanted to put this up to give context, and I'll continue to refer back to how ecosystem function might change because the fungi are responding to climate change, and then they are having effects that can feed back into that as well. Going back to these groups, I had talked a little bit about pathogens. They not only infect plants, but they also infect humans. So one thing I always kind of talk about, especially if I'm giving this talk in Southern California, is, and this is a slight deviation, but I want to mention it anyway because it might be helpful to know, is valley fever is a fungal infection that infects human lungs. It's relatively uncommon, but research is showing it's becoming more common under climate change because as things get drier, remember that image I showed of a dry, dusty desert and that soil is being picked up? This human pathogen actually is able to be both a pathogen, so that red icon, and a decomposer, which is the blue icon. So it lives in the soil, it lives as a decomposer, it likes to decompose proteins, so animal tissue. But when it does get picked up by the wind and lands in someone's lungs, it could be a human, it could be any mammal, so dogs can get it, sea lions can actually get it, dolphins have been seen to get it. When it lands in your lungs, it starts growing in your lungs, so it works as a pathogen. So that's another thing I want to add is some of these specific species of fungi are able to live 2 different lifestyles, so they can be a pathogen and a decomposer. So valley fever infects your lungs. It's prevalent in California and Arizona. We know it's becoming more common under climate change. It's also spreading its range. So it's known to cause disease all the way up far north as Washington and as far east as Texas. So if you ever come down, and I used to give this presentation before COVID, so it's unfortunate that it's during this, but if you ever come down with flu-like symptoms that don't go away for months or weeks sometimes, and people have a hard time diagnosing it. So if you have that and you have a negative coronavirus test, it could be valley fever. So one of the biggest challenges with dealing with valley fever is it is often misdiagnosed for a long time because doctors they're not always aware of it. People aren't always aware of it. So this is a little bit of a deviation from my project, but I always like to put it out there if I'm presenting in Southern California is if you ever feel sick, it can be flu-like, difficulty breathing, and it doesn't go away. It could be valley fever. So always ask about that. Okay, back to the regularly scheduled program of fungal pathogens. So they infect humans. They also infect plants. Another reason that it's so important to study how these fungal diseases move in the air is because we actually lose tens of billions of dollars in crops in the US just from fungal pathogens. So this is an image of wheat rust. It affects most of the wheat belt in the center of the US. So from our border with Mexico to our border with Canada, in the interior of the country, that area grows a lot of wheat and they lose a lot of crops due to fungal pathogens. And you can imagine if that whole area is suffering from wheat rust, that the pathogens moving by wind is probably a big role in that. So it's really important to understand how these 3 different groups of fungi move in the air. It's starting to be a more studied topic as of recently, and I'll explain more why that is later. Another reason why fungal pathogens are a threat to crops is we use fungicides to treat them. And we're having the same issue with fungicides that we're having with antibacterial compounds is we use antibiotics to treat infections. We often use them too much or don't use them the right way. And we're starting to see diseases that are resistant to antibiotics. And it's the same thing with fungicide. We're using this fungicide to treat a bunch of our crops. Unfortunately, we're starting to see some species are becoming resistant. So again, there's many reasons outside of ecosystems that contribute to the importance of studying this. So I'm going to jump into one of my research projects that's going to cover how fungi disperse in the air and how does that relate to ecosystem function, because we really don't know that much about who's dispersing through the air, who can travel the furthest. We actually don't know much about it at all because it's very hard to study these things that you can't see. So I'm going to walk you through a couple of questions for this first research project, like what are the environmental factors that structure communities? And when I talk about fungal communities, the analogy for that would be the types of plants that we find at Cabrillo are different than the types of plants that we would find more inland. So there's going to be slightly different species. There might be some overlap in species, but they're going to be slightly different. The abundances, how many of those species between Cabrillo versus Anza Borrego, for example, are going to be different. So we kind of want to understand how environmental factors structure different communities in microbes, just like we understand how that works with plants. You know, a coastal environment has very different environmental conditions than a desert, and we see different plants because of that. And we want to understand that for microbes too. And pardon the jargon for these question slides. I'm just gonna, if you listen to me, I'll explain it using more straightforward words. So I apologize for not rephrasing these in advance. So then how are these 3 different groups I talked about distributed in the soil versus the air? So we're gonna see different amounts of those groups in the air, different species, and we want to understand who are the fungi that are spending more of their time in the soil, and which fungi are actually able to travel far distances in the air. And then how does that affect how big of a range they have? What area can they cover and live in? And part of that is just going to be a function of their spores can travel that really far distance. So to answer these three questions, I guess 5 years ago now, I took a very long road trip armed with a shovel and an air sampler and lots of dry ice. And me and my collaborator drove across 5 states, so California, Nevada, Arizona, New Mexico. And we took soil and air samples at each of these 60 sites to get the fungal communities in both the soil and air at these sites. And probably the first thing you're wondering is, how do you take an air sample? No, I do not run around with a trash bag trying to capture a bunch of air and store it. I've been asked that before. We have this little machine that actually uses a pump to suck in volumes of air, and the particles in that air then get forced onto this little petri dish of agar. So jello-like material in a petri dish. The fungal spores get pushed using the pump into that agar, so we're able to collect them that way. So this air sampler, because we were covering such a large distance, we sampled 100 liters of air, and this is a relatively small amount. So you'll see in the second project that we did much larger volumes of air, but that's to give you context of how much air we sampled. And then for soil, you just essentially dig it up and put it into a tube, and you store both types of samples on dry ice, because microbes, we want to preserve the microbes as they were found. We want to keep that community as it was in the environment, so we don't want different microbes growing as we store them at room temperature. So we basically put them on dry ice and kind of freeze them in time so we can capture that. And there's a little DNA symbol, and I will talk about how we are able to identify microbes using DNA in just a little bit. So the first question, I talked about environmental factors that we wanted to explore as influencing the fungal community. So we used some spatial factors, some climate factors, and some soil characteristics. So spatial is just where you are and how high up you are. Climate is temperature, how much rain do you get, and vapor pressure deficit is essentially humidity, and we'll touch on these later. Soil characteristics include how acidic it is, how moist the soil is it really packed, or is it kind of sandier, and soil salinity is the last characteristic. So we collected all of those on our road trip, and then a lot of the work for microbial ecology is done in the lab. And the reason for that is we just can't identify and count microbes in the field, right? So if you were doing an animal survey, you're able to go out and follow protocol and count the animals, identify them, record that data. You would go out many, many times to get enough data points. You just can't do that with microbes. I wouldn't be able to go out in Cabrillo and just like tell you that, even if I brought a microscope with me, because there's so many of them. Many of them, things that are microscopic can still range pretty greatly in size. So it's very, very difficult to do it. So unlike animal or plant surveys, we have to bring back these samples to the lab to figure out who's there and how many of them there are. So the same types of information we would think about plants and animals, we do with microbes. And so these little icons here are just showing those 3 different groups in a little community. So on the left, you'll see a community with an equal amount of pathogens, decomposers, and symbionts. And then that other community is a little bit more pathogen dominated. So you'll see me use that to kind of indicate different types of communities. And this is sort of like the difference between, again, the plants you would find at Cabrillo versus the plants you would find at Anza-Borrego. So once we bring back the soil and air samples to the lab, I'm just going to do a quick summary of what we do to get this information. We essentially get the DNA sequences of all of these microbes, and that's what we use to identify them. So we're able to do that With humans, right, we do the sequencing of your microbiome, or the human genome is a more common thing. So we essentially do that with the fungi we collect. We actually, so the trade-off between not being able to see them is the methods we use allow us to process millions of DNA sequences. So remember I talked about that tiny amount of soil we collected and those 100 liters of air on a Petri dish. All of those samples put together literally generate millions of DNA sequences. So you get a lot of data for how much work you're putting in. So even though you don't get to see them, it is pretty cool that you're able to get so much information. And this work is all done on the computer. For context, I definitely did not print out a million spreadsheets with DNA sequences and identify them. We have great programs and coding systems you can use to kind of do this in a manageable way. And what that gives you is a list of all the fungal species that are in your samples, how many of each species were found, and then in which samples you found them in. So to give context for this, at Cabrillo, we have, the different reptiles we have. There used to be around 17 species. Now we find 10 or less different species in Cabrillo. And to give you context, the number of fungal species I found in this study was 3,000. So there's 3,000 species we're dealing with. And we see how many of each species there are and in which sites. And then you sort those species into these 3 different groups. So you can assign them a little identifier, like this fungal species is generally a pathogen, this one is a symbiont, this one's a decomposer, or sometimes this one is a pathogen decomposer. So we assign them an identity, and these identities are based off of studies. So there's 3,000 species, but there's studies to back up if you can assign those species into one of these 3 groups. And we can't do that for every species. There's so much research in the world that needs to be done, but we're able to do it for about half of the species we find, so 1,500 species. So one of the first questions were which environmental factors kind of contribute to the different communities we see. And so these were the factors. And in the soil, the most important things are where you are, as well as the temperature you see, the amount of rain you get, and the humidity, as well as how packed the soil is. So these things make sense. We see this with plants too, right? There's an effect of how much rain you get and what the average temperature is. So this is very similar to things we see with other organisms, and that makes sense. That's what you want to see, is that these things are kind of following what we know about biology. So I've listed, the underlined factors are the ones that are most significant. There's about 7. The factor that explains most of the variation or most of the differences we see between these communities for fungi in the soil is humidity. So more humidity is less stress in this case on the left, and less humidity is more stressful because all living things need water to survive. Microbes often get their water from the air. And so if you have more stress because there's less water and it's less humid in the air, you're going to see different communities compared to fungi that have more water available to them. And I did the same thing in the air. So we used the spatial and climate factors. And the most important thing for fungal communities in the air is how much rain you get and where you are. And the factor that explains how those communities look the most is actually the rain. So you'll see differences in areas that receive more rain versus less rain. So this project really wanted to, my main goals for this project was to see between these 3 groups, which ones are more in the soil, which ones are more in the air, and how does that factor into how far apart you can find these. So in the soil, decomposers are the most abundant group. And this completely makes sense, right? When you're decomposing things, they're on the ground. Either leaves have fallen from the ground, a plant has died, it's on the ground, a tree has fallen over, it's on the ground. So it makes sense that decomposer fungi are the most common. followed by pathogens, and then symbionts are the least abundant in soil. And I do want to note that if we had taken these soil samples right next to a plant or right next to a tree that is known to have a relationship with beneficial fungi, that green bar would be a lot higher. But we wanted to kind of sample these sites randomly and get an idea of what's in random patches of soil. And the same thing for the air. We did the same thing for the air. The results are different. Pathogens are by far the most abundant group in the air. So you can see they have the most. They are clearly able to disperse their spores really well in the air if they're the most abundant group in the air. Decomposers are next, and symbionts actually have a very small presence, which again makes sense because they form partnerships with plant roots. So they're going to be actually traveling through the soil to find other plant roots to kind of colonize and grow with. I wanted to see if their abundances in the air and the soil also matched with their range in the soil and air. So what I did for this was calculate the distances between every site to each other site. And then for each of those 3,000 species, you find the two sites that are the furthest apart, and then you get that distance of what it is. And that is used as a metric, a way of measuring range. So how broad is the area you can find them in. So range is measured in kilometers here, from 0 to 1,000 kilometers. And we get the range for each species of fungi, and then we average it across all the species that are considered pathogens, and all the species that are considered decomposers, and all the species that are considered symbionts or beneficial fungi. And so what you find is pathogens have the broadest range in the soil. And this kind of makes sense because if they are really abundant in the air, they can travel much further than a fungi that is relying on physically growing through the soil to disperse. Some fungi get consumed by insects or rodents, and then whenever that rodent or insect poops later, that's where the fungi lands. So they're traveling further distances, but still pretty small distances. So pathogens that are traveling in the air can travel just way further, and we're seeing that reflected in the areas they can grow in the soil. This is followed by decomposers, and then the beneficial fungi or symbionts have the smallest range. And I also think part of the reason why symbionts have the smallest range is their partnerships with plants can often be very specific. So they're probably also just following where the plants that they form partnerships with are. So they have a smaller, tighter range. And there are no patterns in the area that these different groups are found in the air. So these bars look like they're different amounts, but when you run the statistics on them, there's no significant difference with the range found in air. So they're traveling large distances, but the pathogens are much more concentrated in the air. And that clearly, or that has a difference in how successful they are in colonizing the soil, as you saw before. And so actually I should point out that those letters just show significant differences, and this one doesn't have letters. So these bars, even though they look different, are actually statistically the same. So the takeaways from this project is pathogen fungi, so disease-causing fungi, are more air dispersed than the two other groups, the decomposers and symbionts. They have a larger range in the soil, and so this is indicating that they may be able to disperse further and faster than other groups because we see them more in the air, and they do have a larger range in the soil. And what that means is under climate change, these disease-causing fungi may be able to move to a better environment that's better for their needs quicker than the other groups. This is true of plants and animals, right? They have kind of a suitable environmental condition that they need to be met to be happy. And as climate change shifts where those are, the plants and animals will also shift in their range too. So studies have shown that plants and animals actually are moving towards the poles. So in our hemisphere, they're moving northward a few kilometers north every year or so. So their ranges are, or sorry, every 10 years. So their ranges are shifting. Microbes might be able to do that at a way faster rate, especially the pathogens. So the speed at which they can kind of move to a new environment under climate change and the extent that they can cover might contribute to pathogen spread in the future. And that's something that many different researchers are working on understanding right now. All right, I'll take some questions on this first part of the presentation.
Awesome. We have a couple of questions. The first one was from Patricia. She referred to an issue that you touched on before. I didn't quite catch it. Patricia, are you on the screen right now? I'm trying to see if
I can be.
Awesome. You want to ask your question?
Hi. I seem to recall that we are having issues here, you know, at the park and everywhere in Southern California with laurel sumac being affected by some kind of microbial pathogen. Do you know about that?
I don't know.
Okay. All right.
But I can look it up. We can talk about it the next time we see you.
Okay. I had a second question too regarding the greenhouse, and I don't know if I should save it for later or now, but when we run into, you know, fungi over there, do we, how do we know if it's bad or good? And I'm asking that because we did replant some or repot some agaves, and there were some very interesting growth, you know, around the roots. But we couldn't figure out whether it was bad or not, and whether to do any kind of treatment or not. You know, we were kind of at a loss there. Some of the plants seemed to be doing fine with it. Some of the plants looked stressed, and it could have had nothing to do with it. We don't know. But maybe that's something that you can let me know. Or is there a system in which, you know, you can identify, or someone can identify that? I took pictures and posted on iNat, but you know, there was no ID or anything on that.
Yeah, the easiest way is to take a little piece of it and look at it under a microscope. But if it's pretty thread-like on the roots and it's not like slimy or anything, it probably is mycorrhizae, but we can take a look.
It was more like little balls, little clusters of balls.
Okay.
But anyway.
Yeah, we should definitely look at that together.
Okay.
Awesome. And then Sam asked a question, I'm trying to scroll up to find it. She said, she said, think of lichen, it's often on rocks or non-soil substrates. Are the fungal symbionts in lichen soil travelers?
That's a really good question. So lichen was some of the species included in the analyses I did. They actually disperse through air. There was like this study done in, I think it was the Sierra Nevadas, where you can kind of see downwind of different lichen patches, the same lichen genetics downwind of it, but not around it or upwind, I guess. They are actually, at least the ones in California, they actually disperse through the air. That's what it's thought to be, which was unexpected when I learned about that.
Awesome. And then Scott Bergen has asked, we've got some more questions coming in. He said, are the ranges in migration of symbiotic plants in response to climate change limited based on availability of symbionts?
Like, so if plants that rely on symbiotic fungi are limited because they rely on those fungal partners?
I think that's what he's asking, yeah.
Yeah, that's a really good question. There is a good study. Let me think about how to summarize this. Yes, so in certain areas, the plants are moving northward. Sometimes the plants move northward and cross into a different soil type that the plants are able to grow in, but the fungi aren't. It's not the right soil conditions for them. So they don't travel with their fungal partners. And I don't think those plants do as well. So you will see areas where there's like a change in the soil, as the plants move north of that, they kind of move a little bit past it, but they stop pretty early on. And that's just going to be a common problem under climate change, unfortunately. If you think about a mountain, as you move higher on the mountain, that mountain is pointed and there's less area as you move higher up. So it's a common problem where, yes, things can move to adapt, but sometimes there's a barrier, whether that's a soil type or just the amount of space because you're near the top of a mountain. So yes, it does limit them if they don't have their fungal partners, and the fungal partners can't go.
Awesome. And then Amy, I see you've typed in a question. It's got some arrows. I'm not sure how to go about asking this question. Do you want to go ahead and unmute yourself and ask Lihn Ahn directly?
My question is if there are places where there's more soil to air transport of fungi versus air to soil transport. So like if there are places where it's more like going up versus going back into the soil.
So I know the answer to the first part of your question. I actually don't know the answer to the second part. There are areas where there is more soil to air transport, and that tends to be the drier desert areas. And I'll actually touch on that on the second part of my presentation. I actually don't know the answer to more air transported fungi landing. I'm sure there are patterns with that. I know there are certainly patterns with what's called wet deposition, which is essentially when it rains, all of those droplets are flushing out all the particles that are in the air. So I would say where it rains a lot, you probably are getting a lot of things dropped from the air. I don't know how that works for Southern California specifically.
Thank you.
Awesome. Great question, Amy. And Jeff wants to know if you can address the issue of bare root fruit trees that are sold at nurseries where the fungi are removed.
I'm not familiar with that issue.
Okay.
Yeah.
Okie doke. I think that's it for questions. If you guys have any more, go ahead and keep typing them in here. We'll have a longer Q&A session at the end where we can capture all of those questions.
Cool. Right, moving into the second part of this story. This is an image of a brush fire north of LA in 1948. It's being fanned by Santa Ana winds, which I'm sure many of us know what they are living in San Diego. It's when we get those hot, dry, windy days in the fall and winter. It's kind of an unusual thing where for 3 days it's like really hot. And what's happening is those Santa Ana winds are blowing from the desert. So you'll see the smoke is going out over the ocean because those winds are blowing from the desert. And we get a lot of fires during the Santa Ana winds because it's so dry and so warm. It's the perfect conditions for a fire to start and to spread because the wind can push the flames. Those same winds are strong enough to topple large trees. So this is also in LA County in 1955. This 75-foot tall cypress tree got blown over by how powerful the winds are because they're from the desert, but they're channeled through these mountain passes, especially in the LA area. And we're still having these problems today, right? We still get Santa Ana winds. They can still be very powerful. And under climate change, it's thought that the Santa Ana winds will be less common, less frequent, but still very intense. So much like the rain that I was talking about. It's happening less, but it's very intense. So just under 10 years ago, the Santa Ana winds were the strongest that have ever been recorded. So sustained winds at almost 100 miles per hour with gusts up to 167 miles per hour, which is pretty serious. I grew up in Florida, so we have hurricanes, and I'm impressed by these numbers. So it's not good. During this really strong event, LAX had to ground all of their planes. The area it was just declared an emergency. You had like concrete traffic lights falling over. So we do get pretty strong events. I don't think they're as strong in San Diego. I just moved here a year ago, so I've only seen one season. I think the way the mountain passes are here, it's not quite the same as LA, but we certainly get these Santa Ana events. So this is the first image I had up. It's a satellite image taken by NASA in 2002, and Cabrillo National Monument is dead center. So you can see Coronado Island, you can see the little peninsula. This image extends as far north to the LA area, so you can see the Palos Verdes Peninsula. There's Catalina Island is that top island. San Clemente Island is that one right below it. And then this extends down to Baja California. And what you'll note is there are these dust plumes and smoke plumes coming from the mountain ranges out over the ocean. So we can see dust containing fungi being pushed out over these coastal areas. So normally wind blows from over the ocean onto land. We have these nice onshore winds that give us our awesome Southern California climate, which is why we pay so much in rent here. Everyone loves it, the weather's great, that's normal. Santa Ana winds are a reversal of that. So as I mentioned, these winds blow from the desert and they're accelerated through the mountain passes. And then as the air drops from the tops of the mountains down to the coast, there's this phenomenon that causes the moisture to be sucked from that air and the temperature rapidly rises. So this usually occurs when the desert is cooler than the coast or when there's changes in high pressure areas. So these dry, hot winds pass over the coastal areas, bringing dust, dropping fungi. I usually get a lot of throat and nose irritation when these Santa Ana winds happen. I know that's really common. So that's part of the reason why. And I was curious after doing this other project about what fungi were being brought in on these Santa Ana winds. They're picking up a lot of dust from the desert. They're traveling really far and really fast. So I was curious what we would see. And I thought we would see a lot more pathogens during Santa Ana winds, right? We see a lot of irritation. We know that there are pathogens that live in the soil that are getting picked up by these winds. So I was really curious about that. So I asked a lot of the similar questions from that first project. So what environmental conditions are contributing to the different communities we see during Santa Ana winds, and versus normal conditions where we have that nice onshore wind and we have a great climate. So I'm comparing the airborne fungi between those two different weather conditions. And do Santa Ana winds just increase the number of spores we see in the air compared to normal conditions? And finally, will we see more disease because of the Santa Ana winds? So to answer this question, instead of traveling many, many places and taking smaller samples, I had a sampler in one place in Orange County. It was a high volume air sampler. So if you look at this image, this air sampler also has a pump, but a much stronger pump that's pulling in almost the same amount of air that was pulled in 5 minutes. With that other air sampler is pulling in that amount in a minute or less, and it's running not for 5 minutes, but often for days. So it's collecting much, much more from the air. So we're getting a lot more resolution with what's found in the air, but we're only getting it at one point, which is okay because we're just comparing weather conditions from Santa Ana's versus normal onshore conditions. So it's housed under a little housing, so we're not collecting the spores that are just falling. We're collecting the ones that are airborne. So you'll see that the air is kind of sucking in air from under that little roof and then putting it onto a piece of filter paper, essentially. And this air sampler is only catching, particles that are under 2.5 microns. So not all fungi have spores that small. And the reason why it's capturing all the particles under 2.5 microns is because at this size or smaller, those particles can get inhaled deep into your lungs and cause health problems. So I was really interested in the ones that would impact our health because I thought pathogens would increase. And another reason why I wanted the small spores is those spores tend to be the ones that travel long distances. There are certainly airborne spores that are generated locally, and those are about 10 microns, so 4 times bigger than what's being captured by this. But I didn't want to get what was being locally dispersed. I wanted to get what was coming in from these Santa Ana winds. So those are the 2 reasons the spores that I'm going to talk about for this study are smaller. So I had 15 samples from normal onshore wind conditions. I only had 2 from Santa Ana winds for the year that I ran this. It's a little bit small for sample size that's typically used in a study, but we do get a lot of data in terms of number of species and number of DNA sequences. So some of these patterns are still statistically significant. The main thing you need to take away from this graph is there is no standard Santa Ana definition at all. So these definitions are often kind of specific to where you are locally. And so I set up a definition for me in Orange County for what I would consider a Santa Ana. And this graph is just basically how I calculated it to make sure the differences were significant. The environmental data I took was weather data every 30 minutes. So temperature, relative humidity, wind speed and direction, and how much rain we were getting. And I used 2 different methods to explore our questions. The first one with the DNA symbol is exactly what I did from the first study. The second one is actually counting spores under a microscope because I wanted to actually see if abundance overall of what was being, like overall of all the spores of the entire community was increasing during these events. So limitations of DNA sequencing, with what I was doing, you can't count the total amount of those communities. You can only do the relative abundance compared to each other. So in this case, we had to count the number of spores, not identifying them, just counting the number of spores under a microscope. And the hemocytometer is actually just a grid that you use under a microscope to count white blood cells. But we just use that for fungal spores. So a lot of these techniques were originally developed for health applications. Sequencing of microbes was definitely developed for human health before cool ecological questions. And that's part of the reason why microbial ecology has become such a large field in the last 10 years and will continue to be a large field as we finally have the techniques we need to answer some basic ecological questions that we already know for plants and animals. Right, so that top one, we did the same thing as the first study, put them into the 3 different groups of pathogens, decomposers, and symbionts. Another thing we did to double check that the samples we were getting are actually coming from different places was use this model that NOAA has, so the National Oceanic and Atmospheric Administration. They have a model where you can tell them where you found a particle, and then it will kind of backtrack from there using weather patterns to tell you where it came from. This is not by no means 100% certainty, but essentially you can see during Santa Ana winds, the stuff we're seeing is coming from the desert area and then landing along the coast. During normal onshore wind conditions, you see things coming south along the coast and then coming in. And the further back on this arrow you go, the more ambiguous it is. So I would focus on the first 2 little arrow points on those. So we see things coming from the southern part of Nevada for Santa Ana winds, and then off the coast of California for normal onshore wind conditions. And these arrows show the path from the 2 days before it landed, so 48 hours before, which gives you a good sense of how far things can travel in 2 days. I think it's pretty interesting. If you don't usually think about those things, it's impressive how far it can travel in 2 days. So the first question, what environmental conditions are driving different communities between Santa Ana winds and normal wind conditions? So these are the factors. And the main difference is whether a Santa Ana wind is occurring. So using that local index, And relative humidity also structures what we see in the air. I didn't simplify the next graph too much, but I will explain what's happening. So we're seeing different communities between the normal conditions and the Santa Ana winds. Basically what you need to look at is the 2 Santa Ana samples I have are in orange. The rest of the samples I have are in blue. And these dots just show each of the communities and each of the samples I took. And they're in this space, which essentially collapses all the differences between these communities. So who's there, how many of them there are, into simplified dimensions. So this space just represents how similar or how dissimilar these communities are. And if you run some statistics on these circles kind of encompass the communities that are statistically the same versus different. So the Santa Ana winds are separating out from the onshore winds. That's the main takeaway from this. So sorry I couldn't simplify it, but that's essentially what it's showing. My second question was, do Santa Ana winds bring a lot more spores just overall? Is the abundance higher? And using that hemocytometer under the microscope, yes, they do have significant differences between abundance. So during Santa Ana winds, we're seeing about 2.5 times as many spores in the air as we do over the ocean. And the question I really wanted to answer was, are we gonna see more pathogens during Santa Ana winds? So the normal distribution we see during onshore wind conditions are kind of half and half pathogens and decomposers. You can see that this amount is the relative abundance here. And during Santa Ana winds, we actually get way, way more decomposers than pathogens. So I did not get an expected ending to this story at all. And I'll connect this to what I think may happen in Southern California and in areas like Cabrillo National Monument. But if you look at this, during Santa Ana winds, we're seeing way more decomposers being brought to Southern California. The amount of pathogens looks like it's going down. It's actually, when you take into account the overall abundance, what we found using the microscope, the total amount of pathogens is identical during normal conditions versus Santa Ana conditions. The same total amount. The decomposers, however, we're seeing a increase of 5 times more decomposers landing during Santa Ana winds. So again, this is summarized here in the takeaways. 2.5 times more spores coming from the desert to the coast during Santa Ana's. Decomposers are increasing 5 times, so both their relative and overall abundance are going up. Pathogens, exactly the same. So what does this mean for Cabrillo National Monument? Jumping back to some of the things I was talking about before, we're having more decomposer fungi from the desert coming to our ecosystems. We're seeing warmer and drier conditions as climate change continues in coastal California. So we're actually getting these fungi that are adapted to the conditions we're going to see moving forward and in the near future at Cabrillo National Monument in San Diego. So we're kind of getting fungi that are adapted to what we're hoping, or not hoping, what we will see moving forward. This could be good news, and I just want to clarify that I'm focusing on one mechanism right here in a very, very broad picture of how ecosystems are going to react to climate change. So we don't know what the overall result will be. And there are literally tens of thousands of scientists working on answering this question from different angles. But what I have to contribute is there's a possible reduced impact in the amount of CO2 we're going to see under climate change because, as I mentioned before, decomposers release carbon dioxide as they break things down. There are many studies that show that as things get warmer, the decomposer fungi release more carbon dioxide because they aren't in their optimum for breaking things down. So the metabolism isn't operating kind of in the way it's used to. So they sometimes release more CO2 under climate change, and that kind of feeds more into climate change, and then you'll get more decomposers releasing more CO2. But if we get decomposer fungi from the desert who are already adapted to this type of temperature and dryness, we might not see that positive reinforcing cycle with more carbon dioxide produced from decomposers in San Diego or at Cabrillo National Monument. So it was really interesting and unexpected to find out that we don't see an increase in pathogens in Santa Ana winds. That's good news. We will see more decomposers arriving, and maybe that will mean our ecosystems will have that tiny leg up under climate change, at least when it comes to breaking down dead material. All right, that's it for me with my 2 projects. I will take questions on this second project.
Awesome, thank you, Lihn Ahn. I don't see any questions in the chat, so if you guys have any, go ahead and just unmute yourself and ask away.
Hi, Lihn Ahn. I'm Deborah. I actually did put a question in the chat earlier for the first part.
Oh, I'm so sorry.
There were a whole bunch of questions in there then. I had a question back to that valley fever. And I was wondering, does it create like a bronchial cough that continues with you for a period of time?
Yes, you can have a cough that stays with you for months, and it can, yeah, weeks to months. People's reactions are all different. So for some people it's very mild, others it's pretty serious in your lungs. And for some people that might be immunocompromised, or if you're pregnant, or if you come from a high risk group, sometimes fungi actually not only grows in your lungs, but in your body. So it becomes spread out. And you can have a cough. I will add, especially since this talk is happening during a pandemic, you can't get valley fever from someone else coughing. There's never been a case of that happening.
Okay, great, thanks.
Anyone else has any other questions, they can just go ahead and unmute themselves and ask.
Hi, it's Scott. It's not quite related to what you were just talking about, but it made me think as a TPERT volunteer about marine fungi and if there's any local symbiotic relationships or pathogens that are affecting other organisms in the tide pools.
Yeah, marine fungi are pretty interesting and uncommon. And I would say that's actually one of the newest areas of research in fungal ecology or microbial ecology broadly is marine fungi. We're still at the stages where we're just figuring out who's there and if they do anything that might be pathogenic. So I don't have a good answer for that. It's a very new field. I will say when it comes to marine microbes, there are diseases that are dispersed through the water that can affect organisms. I guess I'm thinking more of coral reefs, so I don't have as much information about the rocky intertidal. I will say microbes that live in the ocean can very easily be aerosolized from waves. And this came up in, I think it was the all VIP call with the red tide and that being toxic. And what I had said was usually the red tides we see off of Southern California aren't toxic. And then like a couple of days after I said that, a news article came out saying, this red tide might be a little different. People are seeing really interesting things, especially at Scripps. So there is thought that this tide might have some toxic compounds. And that's gonna affect people more than the tide pool organisms. One good thing to know is as those compounds get aerosolized by waves, the concentration of those aerosols and compounds dramatically decrease with elevation. So if you're standing on the cliffs by the tide pools, you are getting way, way less exposure to whatever's being aerosolized by the waves than if you're down at sea level. So that's actually something I'll mention at the tide pool committee meeting tomorrow is if you're worried about that, just stand on the cliffs and that'll be a really, really good mitigation just in itself. The other thing, sorry, this wasn't directly related to your question, but we have wastewater treatment plants in various areas in San Diego, and they're supposed to be dumping, pretty far off the coast. However, in the Tijuana River estuary, we often get sewage from untreated sewage from Mexico coming through, especially when there's heavy rains. So I do monitor for wastewater microbes. So coliform bacteria is monitored in the tide pool area. And of course, I will let you guys know if that ever becomes at a higher level than what should be there in the tide pool area, because this is a new research area, but there's a lab, the lab I came from before this is studying if microbes that are in the ocean can be aerosolized by waves and then cause health problems. So very, very new research that I'll be sure to share with everyone once it comes out.
Awesome. And then I think Sam said, will that be filtered out by masks? As in, should we be wearing masks when we're in the tide pools?
For the toxins, yes. That's a good question. It depends on what the toxin is, which I don't think they've figured out what it is. There are some toxic compounds that are concentrated in the bodies of the dinoflagellates that would be aerosolized in bigger particles, which depending on the mask you're wearing, it will get filtered out. I would say cloth masks will filter out some of it, but not all of it. However, if the toxin is a compound that's a gas or a volatile compound, which it's just that molecule floating in the air and it's not a bigger particle, I would say at that point, no mask will filter that out. And that's a topic that's being researched now too in San Diego, but there's no data on that yet.
All right, any other questions? I'm trying to keep up with the chat as much as I can. Any last minute questions? If you want to unmute yourself or type it in if you're shy.
While we're giving time for people to maybe ask a last question, I will just put this up. If you do think of something that comes up after this related to microbes, feel free to email me. It's posted right there. I'm happy to answer any questions.
But we do have one question from Brian. Sorry to interrupt. I don't know what to make of this. He said, have you ever seen the Nat Geo fungus causing ant zombification? Are human zombies possible?
So human zombies aren't possible, at least with fungi. So the fungi that causes, for people who don't know what this is, there's a fungus that infects insects, and it essentially like takes over the insect, and it gets the insect to climb up to a high spot, and the fungus grows all over the body of the insect, and that insect from a very high spot then dies and releases those fungal spores everywhere. So again, with dispersal, it's beneficial for the fungus to go to a high area so the spores can disperse further and infect other insects. I've never seen that in real life myself. However, I have found that fungus in my samples. So it's called cordyceps, and I've seen cordyceps in really low abundances in my samples. So sometimes they just travel really far. Oh, and they're used, I think, in the Central Valley to control pests. They use that zombie fungus. So I'm guessing that's where it came from in my samples.
Awesome, thank you. Alrighty, if I've missed any questions in the chat, go ahead and speak now. Otherwise, Lihn Ahn, thank you so much for taking the time to compile all of this together and present to teach us about fungi. Fungi? Is it fungi or fungi?
So technically both are correct. That has been confirmed by a fungal person, and I honestly don't even know which one I say most of the time. I probably switch between the two. And I just want to say I appreciate everyone with their cats. I love cats, so it's very exciting to have a cat audience. I think that's a first for me. So hello to all the cats out there.
Awesome. Well, thanks everybody for calling in and participating and asking some really, really good questions. I have recorded, I've triple checked that I've recorded this presentation, and we will be posting it on VIP Voice. And I'm gonna recruit Dan to help me with all of that. It's been a real pleasure to see everyone's faces, and I hope we can do something like this again soon. Otherwise, I think that's it. I hope you guys have a great rest of your day.
Cool. Thank you, Satara. Bye, everyone.
Thank
Thank you.
Thank you.
Thank you.
Yeah.